Thanks. I'm going to be talking this morning about an approach to cancer immunotherapy that has predominantly been used in only certain kinds of cancers, but now I think we understand how to utilize immunotherapy for the treatment of a wide variety of common cancers. And the particular approach that I'm going to be talking about is a direct approach in which we use immune cells as drugs to treat patients with cancer, much as we might use a chemotherapy agent. And you can see a basic outline of this general approach of what's called adoptive cell therapy or cell transfer therapy. And as you can see just reading clockwise on this cartoon, we will excise a tumor. We will, by a variety of different methods, isolate multiple fractions of the lymphocytes that are infiltrating into a tumor. And intuitively, what better place to look for a T cell doing battle against a cancer than within the stroma of the cancer itself. We have ways of identifying the immune cells that can recognize the cancer. We can then grow them to large numbers. We probably have a few times 10 to the 12 lymphocytes in our body. We can easily grow 5 times 10 to the 11 lymphocytes using modern techniques on one shelf of a tabletop incubator. We grow those, and then very importantly, for these cells to have activity in the body, we first have to completely lymphodeplete the patient. It seems paradoxical that we have to get rid of the immune system before we give these cells, but in fact, we need growth stimuli for the cells that we give. And when we lymphodeplete the patient, we see for the first time in the circulation IL-7, IL-15 T cell growth factors that will stimulate the cells that we administer. We each have about 1 million circulating lymphocytes in our body per milliliter in our body. We don't have 10 million, we don't have 1/10 of a million, we have 1 million lymphocytes per milliliter because that's the number of lymphocyte it takes to saturate all of the IL-7 and IL-15 that's made by stromal cells that will stimulate lymphocyte growth. When we get rid of normal lymphocytes, all of these cytokines now act on the cells that we actually administer. Well, this lymphodepleting regimen that's critical. I show you one of our most recent studies just completed of 101 patients looking at a chemotherapy regimen, cyclophosphamide and fludarabine, which eliminates all of the body's lymphocytes for about 8 days before it naturally recovers. We compared that to a depleting regimen that added whole body irradiation. It didn't make any difference. The complete regression rate in patients with widespread metastatic melanoma was 24% in both groups. But especially notice that of the 24 patients that had complete regression of all of their widely metastatic disease, and these were heavily pretreated patients when they received their own cells selected for anti-tumor activity, only one of these patients has ever recurred, and all of the remaining patients are in ongoing complete regression. If we now take all of the trials that we've done utilizing this particular approach in melanoma, 194 patients that you see, there were 23% had complete regression of all of their metastatic disease, and notice of these 44, only 2 have ever recurred, and the remaining 42 patients are in ongoing complete regression and are likely cured. And so these studies demonstrate that adoptive cell therapy can mediate complete, durable, and likely curative, many of these patients beyond 10 years, regression of metastatic melanoma. Now, one of the critical questions that came up is, what do TIL recognize that enables the in vivo destruction of the last cancer cell? And the fact that we were seeing these complete regressions of widespread disease, there was no relationship between the bulk of disease and the likelihood of having a complete response. Sometimes we see regressions happening within the first 2 to 3 weeks after administration. We saw this specific cancer regression in the absence of any off-tumor, on-target toxicities in patients. And that's what led us in the treatment of patients with melanoma to explore the role of specific cancer mutations as the likely targets of these tumor-infiltrating lymphocytes that were administered to patients. Now, I know people in this room know immunology, and obviously, although these mutations are not self, they are recognized as foreign. For mutation to be a cancer antigen, I just want to emphasize that it has to be processed intracellularly into a 9 to 11 amino acid peptide, and that peptide has to fit into the groove of one of that particular patient's MHC molecules. So that's only some mutations, and in fact rare mutations will be immunogenic because, of course, this small peptide has to fit into one of the six class 1 or class 2 molecules of that patient. And so a mutation that might fit into one patient's class 1 molecules might not fit into another patient's class 1 or 2 molecules. And you see here for class 1 and here for class 2. So we developed a technique that we first described about 2.5 years ago in Nature Medicine. It's a blueprint for the generation of T cells that are reactive against mutations in that particular patient's cancer. And so we do this by isolating genomic DNA and RNA. We do whole exome and transcriptome sequences to identify the mutations. And you know far better than I how easy that is to do. We have a NextSeq in my own lab. We can do the sequencing in 3 days. Bioinformatics another 3 days. And so by a week later, we know now every non-synonymous mutation, every mutation that changes at least one amino acid that's present in that cancer. And by doing the transcriptome sequencing, we know which of these are expressed. And the question then is, are any of these mutations actually recognized by a patient's T cells in these patients that had metastatic melanoma with complete regression of their disease? And so the way we did this is we synthesize what we call mini-genes. We take the non-synonymous mutation and we put it in the center of a 25-mer peptide. That is, we flank this individual mutated amino acid with 12 normal amino acids on either side of that mutation. And so you see here this histidine mutation. We've also sequenced the normal cells, so we know the sequence, but we know the sequence of the human genome. So we can put 12 normal amino acids here on one side. We can put 12 normal amino acids on the other. Now this 25-mer has to contain every possible 9 through 12 amino acid peptide, 12 amino acid peptide that contains this histidine mutation. It can either be the last mutated amino acid, so this would be the peptide. It could be the first one, and this would be the peptide. But this 25-mer encompasses every possible peptide that might fit into the groove of a patient's MHC molecule. We call this a mini-gene. What we then do is connect all of these 25-mer mini-genes together into what we call a tandem mini-gene. We do this with the DNA sequence. So we have now each mini-gene connected one to the other, each one encoding a 25-mer peptide. We convert that to RNA, in vitro transcribe. We put it into the patient's own antigen-presenting cell. So there's no predictions involved in this. You can predict often which peptides might bind to class 1 molecules. Predictions, however, for class 2 binding are uncommon, and there are many, given the polymorphism of class 1 and class 2, and especially class 2. There are hundreds of different potential class 1 and class 2 antigens that these peptides might bind to. And so prediction algorithms make this very difficult. If we transfect all of these mini-genes into the patient's own antigen-presenting cell, we let that patient's dendritic cell do the prediction for us. It contains all the class 1, all the class 2 molecules. We put all of these genes into that antigen-presenting cell, and now just co-culture it with the T cells that caused a complete regression of cancer in that patient. So one of the advantages of this approach is that there's no need to do any peptide binding. In one fell swoop, in one assay, we test every candidate peptide and every MHC locus that's present in that patient and is included in the screen, and most importantly, you no longer need the tumor cell line, which has been a major obstacle to making progress in immunology because you can grow melanoma cell lines 60% of the time, but you can grow GI cancer cell lines 1% of the time. You just can't grow breast cancer cell lines, or bladder cancer cell lines. It's extremely difficult. There are a very limited number that exist. But here, we, in a sense, by stringing all of these many genes together, we have an avatar of the entire cancer cell because we have a string of every mutation that is present. Well, what we then do is we then put that RNA into the antigen-presenting cell. We take the TIL cells that mediated complete regression of cancer, put them in co-culture with this antigen-presenting cell, and then look to see if, in fact, the T cell is recognizing anything on that antigen-presenting cell. So here's an example of how we did this in a gentleman in his 20s who had widespread melanoma. He had 71 non-synonymous mutations following his whole exome sequencing. We therefore constructed 12 tandem mini-genes containing 6 genes each, and then we put each of these 12 tandem mini-genes into the patient's own antigen-presenting cell. And notice that only tandem mini-gene 1 is recognized, and this is on the basis of interferon gamma secretion following the co-cultivation with this tandem mini-gene 1. None of the others were recognized. This is the autologous melanoma cell line, which was recognized. Well, that means then that one of the 6 mini-genes encoded by this tandem mini-gene, one of these 6, is the gene that encodes the protein recognized by the TIL that mediated complete regression. Well, how do we determine which one of these 6? Well, what we simply do is just back mutate each one at a time back to wild type so that it's no longer mutated. Well, when all of the 6 genes are mutated, of course, we get recognition by the TIL that caused the regression. When we back mutate this first one, we still get recognition. But when we back mutate this second one, all recognition of this antigen presenting cell is lost, thereby identifying this particular gene, a KIF2C, a member of the kinesin family that's involved in the stability of the mitotic signal, as the exact protein and the exact mutation being recognized by the TIL cells that cause the regression of multiple lung, liver, bony, and subcutaneous metastases in this patient. This is probably the most important slide that I'm going to show today. So we've done this from 26 patients with metastatic melanoma that had widespread disease that were refractory to all other treatments. The median survival of these patients is about 6.5 months. And from these 26 patients, using this tandem mini-gene approach, we identified 69 somatically mutated gene products. Many patients have more than one. We have a paper about to come out in which we recognize 10 separate mutations in a different patient using these TIL cells, and we're getting better and better at recognizing them. But notice that these individual genes are restricted by class 2 molecules, by a whole variety of class 1 molecules, A11, A2, A29. But most importantly, every one of these genes recognized is unique. Every one of these 26 patients with melanoma who developed T cells that recognize their cancer recognize an absolutely unique determinant, and none were shared among patients. And these represent genes from virtually from a very large number of intracellular proteins. And we've now done it on additional patients. Again, we've never found the same gene recognized by a patient's TIL that mediated a complete regression. So it thus appears that adoptive cell therapy can mediate complete, durable, and likely curative regressions of metastatic melanoma based on the recognition of immunogenic cancer mutations. And in fact, more generally, and there are papers that are appearing on this virtually monthly now, it appears that the recognition of random somatic mutations is the final common pathway that explains regression from virtually all immunotherapies that we know about, except the ones where we genetically manipulate cells. Interleukin-2, the checkpoint modulators, anti-CTLA-4 or anti-PD-1 that are becoming so popular now, anti-CD40, and certainly our tumor-infiltrating lymphocytes We finally understand, just in the last year or so, this final common pathway. We finally understand that the immunotherapy of cancer is based on mutation recognition. Well, there are about 8,000 deaths from melanoma each year in this country. There are 585,000 Americans who died of cancer last year, and 90% of those patients died from solid cancers, the common epithelial cancers that start in the linings of our organs, from the rectum, the colon, the stomach, the esophagus, the pancreas, the ovary, the prostate. It's those solid tumors that result in 90% of the deaths of everyone in this country. In fact, the disease is so common that 1 in every 3 of us are going to develop an invasive cancer sometime during our life, and 1 of every 5 of us will die of cancer if we don't find better treatments for this disease. And so the question that we then approached was, can we utilize this approach of recognizing mutations to treat the common epithelial cancers by targeting unique immunogenic mutations expressed by that cancer? Was this something that was to a melanoma, but could it be true for all cancers? One of the first patients we treated was this 45-year-old woman who had a metastatic cholangiocarcinoma. This is a cancer that starts in the bile ducts of the liver. She had half of her liver removed, but as happens 80% of the time, the tumor recurs following surgery. She developed multiple lung and liver metastatic deposits. She went on to chemotherapy, which cures no one with metastatic solid tumors. Very rare exceptions, choriocarcinomas in the men, in women, testicular cancers in men, choriocarcinoma in women. Other than that, we have no curative treatments for any metastatic solid cancer with the exception of melanoma, as I've just mentioned. Well, this patient received cisplatin gemcitabine, progressed, more chemotherapy, Taxotere, progressed, developed a lung lesion that we resected for TIL. We gave the patient the TIL. We did not have an objective response. To have a response by classic oncologic criteria, you have to have at least a 30% reduction in the sum of the longest perpendicular diameters of all lesions. This patient had a little bit of stabilization, but no real response, and then recurred in both liver and lung metastases that were growing quite rapidly. We then utilized this tandem mini-gene approach to see if we couldn't purify cells or identify cells that would recognize unique mutations in her cancer using the blueprint that I mentioned by taking all of her exomic mutations and trying to identify whether her TIL could recognize any of them. Well, this patient only had 26 mutations in her lung metastatic deposit. We made 3 tandem mini-genes containing about 12 genes each, and notice again, tandem mini-gene 1 was the only tandem mini-gene that was recognized. And that was by an ELISpot assay, interferon gamma secretion, following a co-culture. But notice, interestingly, that the T cells in this co-culture with tandem mini-gene 1 were upregulating OX40, which is a molecule that gets upregulated by CD4 cells when they come in contact with their antigen. There was a small upregulation of FO and BB, but mainly OX40, suggesting that this might likely be a CD4 cell that was recognizing this tandem mini-gene 1. Oops. Hit the wrong button, sorry. Suggesting it was a CD4 cell. We then looked at all of the 12 genes that were being encoded in tandem mini-gene 1 by reverting them to wild type, and only this one gene, ERB-B2, when mutated back to wild type, turned out to be the gene recognized by this patient's TIL that had fairly minimal to no effect in treating her when large numbers of these cells were given. And this was true based on an ELISpot assay, but also by OX40 upregulation, thereby identifying this ERB-B2 IP has the mutated gene recognized by our TIL. It binds to ERB2, it attenuates downstream RAS and ERK signaling. We developed a technique now to purify the cells that were recognizing this ERB2 IP. And this now culture contained 88.5% of the cells in the culture now all recognized ERB2 IP. We gave this purified population to this patient, and she's now had an ongoing regression of her lung and liver metastases, ongoing at 2.5 years. And I'm going to show you our X-rays. We first reported this in Science about a year and a half ago. And here's a CAT scan of this patient, a cross-sectional cut through the chest. You can see this large lesion that was growing. This patient had a potential median survival of 3 to 4 months when you have this much lung disease. She had multiple liver metastases as well. As you can see, this lesion is now a scar. This lesion pre-treatment has disappeared. Again, she's 2.5 years later. This is the only remnant lesion, very light on PET scan, and is continuing to shrink. These two large lesions have disappeared, and this lesion is also dramatically smaller and breaking up. And you can see the liver metastatic deposits have disappeared as well, leaving this hole in the liver. Now, this patient had this dramatic response that lasted 9 months before a single metastatic deposit appeared in her left lung. We resected that, as well as a hilar lesion that had not completely disappeared. That tumor was resected, leaving the patient now disease-free, and she remains disease-free. You can see now this next most important slide, and that is with 37 patients with common epithelial cancers. All of these were absolutely unique, with the exception of one lesion that was shared, and we're now talking about tumors from the colorectum, from the pancreas, from the bile duct, from the lung, from the esophagus, the breast, the ovary, the cervix. From virtually any tumor we can identify, or put it this way, from almost 80% of tumors of any type, we can identify unique mutations that are recognized by T cells within that patient. Again, all of these were completely different, with the exception of one deposit that was shared, and that was a mutation of the KRAS oncogene, the most common oncogene present in patients, in cancer in general. Patient 3995 had a mutated KRAS. Patient 4095 also had a mutated KRAS, completely different patients, incidentally both restricted by a relatively uncommon class 1 restriction element, C0802. And the same for this patient 3995. Present in about 8% of Caucasians, about 12% of African Americans. KRAS, because it's such a common oncogene, the most common, is present in 70% of pancreatic cancers, 1/3 of colorectal cancers, 20% of lung adenocarcinomas. Our T cell that recognized KRAS appeared to recognize G12D, which is the most common of the variants of KRAS mutations, which tend to occur at this hotspot of amino acid 12. How did we find it? Again, using this blueprint, studying tandem mini-genes. This was a 49-year-old female who had metastatic colon cancer. She had a sigmoid colectomy. It was a very aggressive disease. It was invading into her bladder. She had multiple lung metastatic deposits. She had radiation therapy to the bladder suture line. She received a very aggressive FOLFOX chemotherapy, did not respond. She had 2 lung metastases that were resected for TIL, and we utilized this tandem mini-gene approach to target her unique cancer mutations. Well, her cancer had 61 putative mutations identified by the whole exomic sequencing. We constructed 5 tandem mini-genes as well as peptides and peptide pools. And again, here's the blueprint that we feel is potentially applicable if we can refine this and identify more antigens and, most importantly, identify the T-cell receptors that can be used for therapy. This blueprint, as you can see, we put all the tandem mini-genes into our antigen-presenting cells. We expanded her TIL, used multiple different cultures, looked at a co-culture to see if her TIL recognized any of her mutations, and sure enough, coincidence, it was tandem mini-gene 1. Of course, we randomly put these into the tandem mini-genes. Our tandem mini-gene 1 was the only one that was recognized based on 4-1BB upregulation as well as ELISpot activity. We purified the population of the KRAS reactive cells, and you can see this lesion has completely disappeared. This lesion is breaking up, which we subsequently resected, and this lesion had no cancer in it at all. This tumor disappeared completely. She had one tumor in her left lung also, which did not disappear, which we resected, and she now is disease-free, and this recurred at about 7 months after her treatment. So she too is now completely free of disease. Incidentally, the lesion, the only viable lesion in this patient, when we removed it to leave her free of disease, did express KRAS, but had lost a segment of chromosome 6, which encoded the CW08 restriction element, which led her, that particular metastatic deposit, to escape. Of course, a T cell cannot recognize a class 1 restricted antigen in the absence of the appropriate class 1 restriction element. So that's a rare event. We see that in about 5% of cancers that can lose class 1 by virtue of beta-2 microglobulin mutation, or in this case, loss of part of the chromosome that encoded this restriction element. Now, interestingly, in most patients that develop T-cell receptors against mutations, we find multiple receptors. So in this patient, we're able to identify 4 different KRAS G12D reactive T-cell receptors that were in the patient's infusion TIL. Now, I've labeled them A, B, C, and D, these 4. 50% of the culture that we gave this patient that recognized KRAS when we purified it, recognized this G12D 9-mer, did not recognize the wild type. For all of the mutations that I've just mentioned to you, they recognize wild type. They recognize the mutation, but not wild type. So you can see it's not recognizing a 10-mer, it's only recognizing the 9-mer. But quite stunningly, this T-cell receptor, or the cells bearing it, disappeared from this patient. We could no longer, by a month, identify any of the seized cells that recognize this KRAS 9-mer in the patient. However, this T-cell receptor, which recognized the KRAS 10-mer and not the 9-mer, was the most highly persistent cell in this patient. And notice, out to 4 months later, 10% of all of the circulating CD8 cells in this patient recognized the KRAS mutation. This receptor, which was 7%, recognized the 9-mer, also persisted, but there were very few of them. And then there was this last receptor. And so there were differences in the T-cell differentiation states, or that might have been the result of this one persisting at 10%, this one not persisting at all. Maybe it was different avidity for antigen, although there doesn't appear to be major differences in avidity among these receptors, thereby leading us to think it was something more intrinsic to the cell that we gave, perhaps this differentiation state, its proliferative potential, that enabled this particular receptor to persist and lead to the regression of the multiple lung metastases in that patient. Well, this is an overall summary now of 61 patients with metastatic epithelial cancers. Our major effort right now, and none of this is published yet, nor is the KRAS material that I just presented published yet. We've attempted to study 61 patients with this wide variety of epithelial cancers, ovary, lung, cervix, as you can see, virtually every one of the common epithelial cancers. And notice in virtually every one, we've been able to find immunogenic mutations. Now, not just mutations, but mutations that are actually recognized by a T cell that we could identify in that particular patient. So 47 out of these 61 patients, or 70% with metastatic epithelial cancers, had T cells that could recognize mutations, multiple mutations in many cases, and we're especially concentrating now on the gastrointestinal cancers, the colon, rectum, stomach, esophagus, pancreas, and 28 of 31 of those gastrointestinal cancers had T cells that recognize unique mutations. And every one of them is unique with the exception of this KRAS mutation. We think we have at least a blueprint to begin to study the immunotherapy of patients with virtually any common cancer, but we need ways to purify these cells so that we can obtain not only the cell, but the T cell receptor from that cell, and we need that desperately for reasons that I'll get to in just a moment. Well, Elena Gross, a postdoctoral fellow in the lab, hypothesized that perhaps there were markers of antigen-reactive cells, and the one that we looked at is a molecule called PD-1, programmed death molecule 1. Now, you're familiar perhaps with PD-1 because PD-1 is the target of the major checkpoint modulators that we use to treat patients with melanoma and smoking-induced lung cancers, about a 17% response rate, and then scattered single-digit response rates for some other tumors that express the target, the ligand for PD-1, which is PD-L1. And so by inhibiting PD-1, we can actually see regressions in melanoma and a small number of other cancer types. PD-1 has been referred to as an exhaustion marker. It's a marker that gets upregulated the more the T cell is stimulated. It's an activation marker. And so we hypothesized that it was the cells inside the tumor that were constantly and repetitively being stimulated by tumor antigen that would most vigorously express PD-1. And so if we looked at all of the CD8 cells inside the tumor, with each dot now being an individual patient, you can see a wide variety of PD-1 expression within the tumors of patients. Very little PD-1, only 3% median in patients in PD-1 in peripheral blood. We had previously published that if you isolate the PD-1 positive cells infiltrating a tumor, they contain all of the anti-tumor activity, but it didn't turn out to be much of a purification. We just published in Nature Medicine 3 months ago in this paper that if we take this 3% of the peripheral blood mononuclear cells isolated from a blood draw or apheresis and isolate that 3%, it contains all of the anti-tumor activity, which is true for the PD-1 positive cells infiltrating into the cancer. You can see we saw no anti-tumor activity against this patient's cancer when we looked at all of the CD8 cells in peripheral blood. None when we looked at the PD-1 negative cells, 97% of them from peripheral blood that was CD8, but all of the CD8 positive, PD-1 positive cells by 2 different assays, 4-1BB upregulation or interferon gamma ELISpot, contained all of the anti-tumor activity. And so now, by doing a blood draw, we could purify the PD-1 positive cells. We can do a whole exome sequencing on paraffin fixed tissue. We can't do the transcriptome sequencing, but we can therefore make all the tandem mini genes we want from the original resection or from a metastatic deposit resection. Take T cells from peripheral blood, and you can see in every patient, and we've done this now in many, many patients, you can see all of the reactivity against tumors present in this PD-1 positive and not in this PD-1 negative population. And there are other co-stimulatory exhaustion markers like TIM3, which are not nearly as good in all patients. It happens to be effective in this patient in isolating tumor-reactive cells, but not in this. PD-1 is a very constant marker of tumor-reactive cells in the peripheral blood. Of course, it contains reactivity against multiple viral antigens or whatever else the patient happened to have been recently exposed to. But we can now take these PD-1 positive cells, as we reported 2 to 3 months ago, and perform our tandem mini-gene approach. So in this patient, 3784 out of 140 mutations. These are just viral antigens, which are used as a positive control. Anti-CD3 just activates all T cells. We see virtually no reactivity against any of the tandem mini-genes, the 9 that we developed. This is the autologous tumor line giving us a little reactivity, not an allogeneic line, but no recognition of tandem mini-genes. We take the PD-1 negatives, no recognition of tandem mini-genes, but now the PD-1 positive, and especially that small fraction of high PD-1 positive cells we could isolate on the sorter, recognize 3 different tandem mini-genes, and again, 3 just completely random genes from the intracellular constellation of expressed proteins, in this case filament A, a different kinesin-like protein, and the DNA binding protein that were recognized, in this case by testing, not by reverting to wild type, but by making the long 25-mer peptides and just pulsing them onto the antigen-presenting cell. We only had 16 that we had to make, and we could identify multiple reactivities in this patient that recognize these random somatic, just bad luck mutations that in this sense convey some ability to react against the cancer. So we have a way to purify the cells, and we're currently exploring the following, and many thanks to Danny Dewick for helping us with much of the DNA sequencing that was involved in this, and especially in this particular project that involved a lot of single-cell sequencing. We hypothesized, this hasn't been published yet, it's just been accepted for cancer immunology research. We took 12 patients in a row and hypothesized that if in fact T cells within the tumor were being continuously stimulated, they should be expanding in the tumor, and the frequency of the cancer T cell receptor might well be an indication of what has anti-cancer activity. And so using a variety of techniques, again, thank you, Danny, for your help with this, we looked at 12 consecutive patients at the top 10 most frequent T cell receptors that were present in that particular tumor just by doing T cell receptor sequencing, but pairing the alpha and the beta chains turned out to be tricky, and a lot of that was done with single cell sequencing. You can see in 11 of these 12 patients we could identify T cells with anti-tumor activity within the 10 most frequent of the T cell receptors in the tumor. And notice that 1, 2, 3, 4, 5, 6, in 7 of the 11 cases, the most frequent T cell receptor in the tumor had anti-tumor activity. And in all 11 patients, if we looked at the top 4 most frequent sequences, we could find T cell receptors that recognized the tumor. Now, these were not all mutation reactive. 5 of the 11 recognized mutations. Some recognized a kind of cancer germline antigen, a kind of antigen like MAGE, A3, or NY-ESO1, that are proteins that are expressed during fetal development but are lost in the adult, but are re-expressed in a very tiny percentage of solid cancers. And so about half were recognizing mutations. And this is our first attempt to use the frequency of T cell receptors as an indication of identifying cells with anti-tumor activity, and we're continuing to work hard on refining this to increase our ability to identify mutations, and this was done by one of our postdoctoral fellows, Anna Placeto. But it turns out that the T cell is not what we want, because by the time we isolate the T cell, it's gone through so many proliferative cycles that it's lost its proliferative potential to a large extent. especially when we start with a tiny number of starting cells. And we know that proliferation of the cells is vital for the success of this cell transfer approach. It's a living treatment, and the cells that we give expand 1,000-fold in the first week after their administration. And so this particular patient who had a colon cancer, a 52-year-old male, I just show 2 slides just to demonstrate. We use this blueprint. We identified his reactive T cells. But what we really needed was his T cell receptor. And so we took those T cells, we subjected them to exposure to the tandem mini gene. There were 2 tandem mini genes were positive. We could isolate those T cell receptors and now put it in a transient retrovirus, put that T cell receptor into a patient's normal cells, and we get completely specific reactivity. You can see this V beta 20 that came from TIL fragment 16 recognizes this mutation SKIV2L, but not the wild type, and this particular T cell receptor recognizes this other gene and not the wild type. And so one of our issues is the isolation of T cell receptors that can recognize tumors. So in this penultimate slide, I've tried to show that cell transfer therapy can mediate durable regressions in patients with metastatic cancer refractory to other treatments. The only patients we see are patients that have been through all standard treatments, many of which are marginally effective. The T cells that recognize unique somatic mutations can be found both in tumor-infiltrating lymphocytes and peripheral blood, and that identification and targeting of mutations unique to each cancer has the potential to extend cell therapy to patients with common epithelial cancers. But here's the problem, and one of the reasons that I was so interested and excited by hearing from Dr. Dueck and having the chance to speak at this important meeting. And that is where to develop personalized T cell receptor gene therapy for patients with cancer. We need to identify not so much the cells, but the T cell receptors in those cells. We know we can isolate, if you just follow me down, the mutation-specific T cells from tumor and blood, but just in tiny numbers. And we therefore have to stimulate them in vitro. After their repeated stimulation in vivo, this in vitro stimulation is required to obtain enough cells for therapy. And that continued replication results in terminal differentiation of the cells and a limited proliferative capacity in vivo. And multiple experimental models have shown that the less differentiated cells are far more effective than the differentiated effector memory and EMRA cells in mediating tumor regression. And so the approach that we're now pursuing is to identify the genes that encode the mutation-specific T cell receptors and insert them into autologous naive or central memory cells that we can obtain from the peripheral blood of individuals. Most individuals in this room contain about 40% of all of their circulating cells are completely naive with naive phenotypes. And those are the cells that can explosively react to an antigen exposure. And what we need is a rapid, safe, and cost-effective approach to integrate these T cell receptor genes into patient PBL. So we need to identify the alpha-beta chain pair. We need a relatively rapid way to insert it into T cells. Right now, we're just making transient retroviruses from a 293 GP cell line, into which you just have to put the envelope gene, RD114, and the alpha-beta chains, and then have a transient retroviral supernatant you can use to put those T cell receptors into patients' own less differentiated cells. And so a current plan, for which we need a great deal of help, is to develop a gene therapy approach for single patient treatments targeting tumor-specific neoantigens. And one of the things we're doing is developing a technique that would be FDA approvable to use transient gamma retroviral vectors that encode these neoantigen-specific TCRs for single patient treatment of solid epithelial tumors, but there are other ways to do it, transposon, transposase techniques. There may be ways with CRISPR-Cas to use AAV to insert these genes. There are a whole variety of different possible approaches to do this. This sounds daunting. After all, what could be more personalized as a therapy than having to use a patient's own cells to attack their own cancer and attack a unique antigen that's expressed in their cancer and nobody else's cancer? It's a daunting approach, but I don't think many patients with metastatic cancers care how complicated the treatment is, so long as it can work. And I think we have here at least the beginnings, as we can improve these techniques, beginnings of a blueprint that we might use to treat many patients with common cancers if we could overcome the problem of isolating these TCRs, pairing the alpha and beta chains, and in an efficient way put it into a patient's own cells to use as a drug for treatment. It's ironic in a sense that many of the mutations that cause cancer may end up being the Achilles heel that enables us to attack the cancer itself. Well, thank you for your very kind attention.
Thank you very much, Steve. We have time for questions if anyone has any questions. I have two questions. I mean, as you just said, your approach really is the epitome of personalized medicine. How do you make it tractable enough, though? Are you going to be able to treat every person with metastatic cancer?
We first used tumor-infiltrating lymphocyte to treat a patient in 1988, reported in the New England Journal. And as we've tried to refine this, there was always a thought that, look, these cell therapies could never enter mainstream oncology. They're too complicated. But in fact, there are now 20 commercial companies. We're working with several of them at the NIH, at least through cooperative research agreements. At least 2 commercial companies, Kite and Novartis, are developing commercialization of T-cell therapy using a common shared gene, CD19, to target patients with lymphomas. There are 20 different companies that are trying to use either targeting CD19 shared antigens on lymphomas, because it's an antigen that's also present on normal B cells, and as you wipe out the lymphoma, you also wipe out the normal B cells, something you can't do for the solid tumors. They're in multi-institutional trials. I fully expect that cell therapy to enter mainstream oncology within the next 2 years. You send the pheresis product to that company, they produce the anti-tumor cell, or for Lion, who are trying, Lion Biopharma, they're trying to develop the TIL therapy for treatment. You send them a fragment of tumor. They then generate the cells, grow them, cryopreserve them, and send them back. And so that's the paradigm now being used by these 20 cell transfer therapy companies that at central locations will receive a tumor deposit or a pheresis product, create the cell for therapy, and send it back cryopreserved. And so Big Pharma has been very slow in getting onto this. Big Pharma likes a drug in a vial on a shelf, so they don't care if they spend $500 million creating the first vial, so long as they can create the second vial for $1. Well, here you have to create a new drug for every patient, and so it is daunting, but it certainly is possible, and I expect the first cell therapies to be commonly used within 2 years.
Thank you. My second question, is the supposed immunosuppressive microenvironment of the tumor a concern to you?
There's a lot of work showing that a whole variety of factors within the tumor itself can prevent T cells from working. There are T regulatory cells, there are myeloid-derived suppressor cells, there are cells that make IL-10, and one of the reasons for the need for this very aggressive lymphoablation, again, it's non-myeloablative, the cells normally return, but we drop every lymphocyte count to 0, white cell counts to 0 for about 8 days before the body normally recovers, and that eliminates all the Tregs, T regulatory cells, all the myeloid-derived suppressors, and many of the suppressive elements, but it's entirely possible that the elimination of TGF-beta or other immunosuppressive factors might augment this activity. And so I think studying ways to impact on the microenvironment might work well in conjunction with this and could be a very important thought.
Very exciting work, and the direction of personalized medicine using T cells and specific TCRs is very exciting. Regarding the slide you had with the first 10 ranked TCRs, just to make sure I understood correct, those 10 first ranks were different in each patient, right? There were no common TCRs in those 10 ranks, I suppose.
Right now, in well over 80 patients, we've never found a shared antigen with the exception of KRAS.
And
all of those were different, every one.
Even if you go lower in the ranks to 100, 1,000, or?
Well, we've only gone down to, we've concentrated on the top 20. But in 11 of the 12, one of the tumor reactive cells was in the top 4. But of those 11 patients, the top 4, only 5 were mutations. Some were other reactivities. Some we haven't identified yet. But when we've identified the antigen, we have never seen a shared determinant with the exception of KRAS. Yes, in the back.
So it looks like you get a complete remission in about 25% patients. In the ones where you don't get a complete remission, what is happening there? Can you see that there's like immunological escape? Is the tumor mutating, and the mutation that you saw was T cells are reactive for is now gone, essentially? And does that also mean that, you know, if you're doing a therapy where you're essentially putting in a single T cell receptor that's tumor specific, is actually purifying the tumor and getting rid of that mutation? And so do you have to actually do like a kind of like an HIV, like kind of highly active retroviral therapy, like multi-pronged approach?
In the 24% of patients that I showed that had complete regressions in melanoma, that was before we were identifying the individual mutations. It was only when we saw that we could cause complete durable regressions of patients with widespread disease that we did the studies that I mentioned and that we published, showing that these T cells were recognizing mutations. So I suspect if we were to go to melanoma and actually identify the cells that recognize mutations, our response rate would likely be much higher, but we've diverted our attention now to these epithelial cancers that, again, result in 90% of all cancer deaths. Now, the question of whether or not the tumor will downregulate expression of an antigen if it's not essential for the malignant phenotype is an important one, which is why we're working hard now to identify multiple T cell receptors from an individual patient so we can give multiple ones at the same time. Because once you have the T cell receptor in hand, it's just as easy to make 5 transient supernatants as one. And my thought is that the reason that patients are not responding is, one, the cells that we're giving are highly proliferative. We have not yet treated a patient with a TCR transduced cell and using cells that are either naive or central memory that have a huge proliferative potential. So a lot of the cells we give are terminally differentiated and cannot work. And again, I think giving multiple T cell receptors at the same time will overcome downregulation of antigens. And we have a paper in press where we identify 10 different T cell receptors, 10 different antigens, mutated antigens from even a single patient. Yes.
We've been using a child's non-ablative regimen from a clinical bone marrow transplanter, and I found the IL-7 and IL-15 also to be very sky high in the first few days. I'm wondering if you have other cytokines that you've looked at that might be important in that expansion.
IL-15 is a really good T cell growth factor. And in fact, we have no circulating levels because, again, 1 million lymphocytes per mil in our blood will saturate all the IL-15 produced by non-lymphoid cells. And so in a sense, the lymphodepletion is just a cheap way to give a lot of IL-15. Now, we get elevations of IL-7, but that's not a growth factor. You can't grow T cells in IL-7. Because at this point now, the patients are depleted. We do see elevations of IL-6, sometimes IL-10, but it's mainly IL-7 and IL-15.
In the part where you showed that most of the specificity was contained in a PD-1 specific cell population, if I remember correctly, it was all on CD8s. Did you observe something similar for CD4s? Did you do that kind of experiment?
We're doing that now, and you can also purify CD4s utilizing PD-1. That's a subject of our current study by Elena Gross, who actually did this in CD8 positive cells. It also is the case for CD4 cells, but we're just developing that information now.
And the second kind of a step further would that be, would you also look in their regulatory T cells in there? Would that be a scope?
When we give TIL, none of these TILs express FOXP3. We do not, nor do they express PD-1, but these can be re-expressed in vivo, which is one of the reasons why we're now conducting a trial in which we give these cells in conjunction with anti-PD-1, even though none of the cells going in express PD-1, nor do they express regulatory T cell markers. We know at least PD-1 can be re-expressed. And we know that because we've given gene-modified cells, so we can distinguish the cells we give from the endogenous lymphocytes, and we see the cells we give upregulating PD-1 after about 2 weeks inside the body. And so perhaps by blocking that interaction, we might be able to improve upon the treatment.
I'm wondering about how high throughput you can make your tandem mini-gene approach. Would it be possible, is there ever a situation where you see, say, 500 or 1,000 mutations, and if so, would it be possible, feasible to screen with the tandem mini-genes that way?
The answer is yes. It's just a lot of work. We do everything by hand. We haven't started using robots at all, but this very much lends itself to robotic analysis. One of the beauties of this is all of the manipulations can be done in a regular laboratory. It doesn't have to be done in a GMP facility. The only thing that needs to be done in a GMP facility is the production of a alpha beta chains of the T cell receptor and the envelope gene that you can then transiently put into the patient's naive or central memory cells. So our entire TIL lab is 675 square feet. And in fact, it got closed down on March 22nd and just reopened on June 17th because we were then told we had to meet full GMP characteristics in the laboratory. But it's very doable. And again, when we recognized the 10 mutations from this one patient, we were screening about 2,000 of these exomic mutations. So we do it in 96 well plates. We're exploring the smaller plates that contain over 380 different wells. But I think there are ways to robotically do this once we get into full swing. Right now, we're trying to understand the biology predominantly.
Thank you.
You noted that there's a paucity of shared versus individual. You did note there are some wild-type antigens, but there are these common ones that have been studied a lot for a few decades now in cancer, these developmental antigens like NY-ESO, et cetera. Is it possible there are some other shared wild-type antigens that just haven't been revealed by this because you haven't searched for them?
Yes.
That's it. I'm just curious of your opinion on that.
These cancer germline antigens are present in low single digits and almost never homogeneously. And we just published this past year, a study of almost 4,000 different common epithelial cancers from tissue arrays. So these cancer germline antigens that are wild type and shared are present in a few percent, and because they're epigenetically modulated, they don't have homogeneous expression. I see very little future for them. We have tried to target some common antigens like CEA, for example, carcinoembryonic antigen, highly expressed in colon cancers, minimally expressed in the colonic mucosa. But the T cell receptor is so exquisitely sensitive that when we treated 5 patients, we got good regressions we treated 3 patients. We got good regression of liver metastases in 1 patient, but we destroyed all of the colonic mucosa by giving even a small number of these cells. Fortunately, it reconstituted, but it was 2 weeks of hell for those patients before we stopped doing it. If you target something that's on a normal cell, these T cells will kill that normal cell just as quickly as a tumor. And so we haven't found shared antigens. 39 years ago, people described monoclonal antibodies, and here we are 39 years later, and it's not a single monoclonal antibody that recognizes a unique determinant that's shared on the surface of cancer cells, possibly with the exception of the EGFR variant 3 mutation in some patients with glioblastomas. Other than that, there are no cell surface determinants that are shared. So we'll keep looking, and hopefully we'll come across other shared oncogenes, but as of yet, we haven't found any.
So, short question, what you've shown so far, that all the mutations that you've identified are unique except for KRAS. Do you think that is caused by undersampling, and is the future perspective that you would like to generate a huge library of all the possible mutations and that you just have to do exome sequencing and pick out mutations that you would like to give to a patient?
Wow, none are shared. And of course, a random mutation can occur anywhere in each of the proteins that are encoded by our DNA. So unless we can find shared antigens, I think we're going to have to find the individual, unique, random somatic mutations. It's remarkable how long it's taken us in cancer immunology and immunotherapy to understand the fact that the immune response against the overwhelming majority of cancers is the result of an attack on a random somatic mutation on virtually any intracellular protein. Really quite astonishing.
Thank you.
Thanks for the introduction, and thanks for having me here. So today I'm going to tell you a project in the lab that we've been experimenting with using sort of a crowdsourcing approach for reusing the increasing amounts of public data that are getting out there. And first, a disclaimer, we're actually not really a tool or database developer per se, but over the years, we've been utilizing more and more public data. And while we do that, by integrating those with some of the data we generate in the lab and try to generate new biological hypotheses, and because my lab is sort of half experimental and half computational, increasingly it became quite clear that the computational folks can really tap into the public data really well, and they can really start to integrate them into their projects, while the experimental people are starting to complain, saying that, oh, I always got to go to XY to really get their help to answer a particular, or to get to a certain data and put them all together and start asking questions off the data. How come there's such a barrier, basically? So this sort of came out of that sort of problem in the lab, and then as it grew, it became quite clear that we would like to make this more publicly available. So just an introduction. So the classical paradigm for doing biology would be you ask a question, right, and you think about an experiment, and then you think about how to do it by yourself. So for example, if you want to look at gene expression signatures of IBD patients with healthy subjects, you want to collect colon biopsies, and then you want to, for example, isolate RNA if you want to look at gene expression, and then you want to do microarrays on RNA-seq, and then you can generate such a data set and start to answer your question. But of course, in the past more than a decade, there's been a lot of data generation, gene expression, of course, but also genetics and methylations and so on and so forth. And the amount of data that's out there, especially residing in sort of different databases, it's really getting quite humongous. And the trend is only sort of getting a bit worse in terms of how heterogeneous it is and how much data there's out there, basically. And really being able to tap into this, I think, meaningfully, it's important. So, a complementary paradigm that's been mainly sort of practiced by computational biologists. For example, Epitope, it's back in the days at Stanford, and also my friend and colleague Pravesh Gottlieb, for example, they would not typically generate any data in their lab, and they would be going into public databases and start asking, can I extract all the existing data sets on IBD? And by looking at information and patterns across multiple data sets, can I derive consistent information or signatures? And then based on that, they would generate new hypotheses to follow up with either more data analyses or in some cases more experiments. So this, to me, it's a complementary approach. You start asking a question by going into existing data and see how much new information you can get and how much of your initial question can be answered by using these data. So there's been quite a bit of publications using this kind of approaches. So for example, looking at drug repositioning using gene expression data, or discovering conserve gene expression blood signatures across diverse types of viral infections. Again, a paper that just recently came out in Immunity from Pravesh's lab. And also looking at sepsis, for example. So as I mentioned, these are the principle behind it is actually quite sound. So even though the data from one particular study may be quite noisy and there may be individual, for example, batch effects, if you can see a signal across tens of studies, it becomes quite intriguing or attractive that you ought to start to follow up in some of the common conserved signals across the different studies. So as I mentioned, typically these efforts are led and performed by expert bioinformaticians. So, the question, as I alluded to earlier, is how do you enable non-specialists to start performing this kind of analysis hands-on, so then they can really directly exercise their biological and experimental intuition, right? So I'm sure there's a lot of biologists here, and you have questions, and you want to be able to basically touch and work with the data. And also I think would facilitate increased interaction with bioinformaticians down the line. So unfortunately, there's really no one-button solution. I wish there's really a one button you can press and then just you ask a question and all the data gets in and you can start to extract information out of it, right? So this is a scenario that you can imagine. You're sitting there, you're a biologist, and you're asking what are the signature pathways that characterize distinct B-cell subsets, for example. Let's say you got your favorite antigen-specific B-cell subset, or what's the common gene signatures among all the bacterial infections. So you can do that now, actually. I mean, there's lots of tools out there. You can go to a public database, you can query the data, you can find the relevant data sets, you can download them, you need to do quite a bit of data QC, typically. You got to normalize the data. You got to, for example, in the case of microarrays, you got to map the probes to the genes. And the other, you got to annotate them and then really design the comparison, for example, right? So there's two barriers. One, as I mentioned already, it's the programming and statistical analyses. That really pose a major barrier for non-specialists. The other, there's a lot of missing meta information. You go into a particular data set, and sometimes the controls label is C1 to C2, C3, and sometimes with C1 October 5th, for example, right? What does that really mean? So you got to go into the paper, look at the supplementary materials, and often you got to contact the author and what do you mean by the C5 with this label? Should I use it or not? Is it a bad sample, for example? So there's a lot of meta information missing. And often you actually want to tap into these data and ask a distinct question, not asked by the original authors. For example, you may have a study looking at a drug trial, and naturally they would enroll a mix of males and females. And by using that information, you can start to derive differences in male versus females, for example. So you want to get that kind of information about the gender, which may or may not be presented in the submitted data. So you want to go back and actually email them. Can you send us a sheet, for example, right? So those information can be sparse. So what we came up with, we try to come up with an approach that's sort of lower those two barriers. So on the one hand, on the more technical side, so we came up with this platform. I called it OMIC. It stands for Omics Compendia Commons, to sort of emphasize the fact that it's a common space for storing and for sharing a lot of these information that I'm going to introduce you to. So we basically pre-processes, in this particular case, is gene expression data. All the data that's in NCBI Geo, we cover about 90% of the platforms that they support. So we run through the existing data through fairly standard pipelines and QC them, so they're already all available. And then we provide a web interface where you can go in and you can search for any data set you like, and then you can start to go in and start to group the samples together in a biologically meaningful way based on the question. Let's say, going back to the gender example, you can go in and you can start to find a study, and you can group the sample and say that these are samples, PBMC samples from females, and you can group another set of samples and label those as males. And now you can start to form these comparison groups. Let's say you want to compare male versus females, right? You can make that, we call that a comparison group pair. So all of that information would be captured by the system as you annotate them, as you move along, and then you can create a compendium out of those comparisons. You can collect, for example, multiple gender comparisons across 10 studies and put them into a compendium, and by clicking a button, it would basically do a meta-analysis for you. Basically, what that means is it goes in and computes all the genes that are different between males and females, and it also looks across multiple studies to give you a conserved signature of male versus females. And all of those are captured by the system. And the crowdsourcing aspect of this basically involves you to click, say, make my groupings, my annotations, and my CGPs public, basically. So then another person who's interested in, let's say, gender comparison, maybe in a slightly different way, may come in and be able to start to reuse what you have done already. And we also try to encourage folks to annotate their groupings and their comparisons using standardized vocabulary so that it's easier to interpret by others. We also have features that hopefully would encourage at least some initial assessment of the quality of those annotations. For example, we encourage you to put in your profile. So we, for example, if you do B-cell receptor sequencing, we know that you likely have some knowledge about B-cells. So if you have annotated and worked with a bunch of B-cell data sets, that makes that a little bit more worthy, hopefully. And there's also tracking of like how often does your particular CGP gets used by other individuals in the system. So that sort of thing hopefully would allow us to provide would allow the system to provide feedback on the quality of those annotations. So this actually just came out in Nature Biotech, so there's some details that you can read about in that paper. So just want to acknowledge the team that really put this together, especially Jason on the left here, who really was responsible for almost all of the website and software development, and Naisha on the upper right, who's responsible for a lot of the data processing pipelines, and Kay, she really started out designing some of the user interfaces earlier on, and Rachel on the ping pong table make all of the videos and in the tutorial. Yong helped with various programming tasks along the way. So just to summarize the basic idea is really we want to entice the biomedical research community. We provide this easy-to-use interface and all the data pre-processed. You can really hopefully go in and start asking questions. And with a few clicks, you can start to get some preliminary results back, and you need to follow up. You can follow up on. And then the hope is, as you use it more, there's more and more of these shared annotations and usable groupings and compendia created by experts in your field, and then that would lead to a more usable repository. So I was going to do a live demo. I'm not sure if I have access to the net here. So this is what it looks like on the homepage, and it's fairly straightforward. You can do searches to search for samples, or you can start from a raw study, or you can search on things that are already available in the system, as I said, annotated by others, and you can also search for these comparison group pairs. So for example, I could search for, so we work quite a bit on macrophages, for example. So we have done form a lot of different groupings from different studies. And you can also click and say, I only want the ones that's already shared, so the public ones, right? So you search again, and then that's what you get back. So just to illustrate what these are, right? So each row here, it corresponds to a comparison group pair. So you have two groups of samples. So for example, if you look at one of them, this is what it looks like. So it's got these raw cells from mouse. It's exposed to LPS for 1 hour, and so these were all grouped together as a sample group. And then there's some annotations provided here, the perturbation being LPS, and it's the time with the perturbations for 1 hour. And as I said, you can put in other things, like let's say, for example, it comes up with these standardized vocabularies, and you can select which ones you like. then you can put them in. And so we encourage you to do it with these standardized vocabularies. And so here, for example, we have two, on the top row, we have two groups, right? LPS at 0 hours and LPS 1 hour. And then the CGP is basically comparing these two, what genes are changing from 1 hour compared to the 0th hour, for example. And you can look at who created it. In this case, this is actually someone that I don't even know. And then you can also look at the usage status. For example, it's used in 5 different compendia, basically, in this case. So that's the basic idea behind a CGP. And now you can look at, for example, you can look at the compendia, and you can look at, for example, let's say this is one that I had. This is looking at vaccine responses, and you can basically group these into this one compendium, and you can, for example, click on meta-analysis, and you can just select them all and say run meta-analysis, and then it will give you results back. And like I said, by using a few clicks, once you got these things all put together, you get your initial results hopefully in minutes, and there you can now really start to think about the biology and start playing around with next step hypotheses or even experiments if you really trust the results. So that's the gist of it, and the tutorial, I encourage you to really look at it because we have very extensive information on both how to use this approach. So we have lots of videos. So Rachel has generated lots of really nice videos, and also some background information on the statistics and the considerations that you need to think about before doing this kind of you're using the public data and also doing meta-analysis. Any questions so far? Yeah, John? Yep. I thought you were raising your hand. Okay.
I guess the first one, I don't know if I should ask them now, but what's your source of the controlled vocabulary that you said for the
mesh terms?
Mesh.
Yeah.
Have you considered doing something actually more?
We did. We actually spent a bit of time in looking into this approach called MetaMap. It was developed by I think the National Library of Medicine. So SNOMED and all these different ontologies. It turns out that it's a bit too complicated. So the amount of synonyms and it's so extensive that you type in something and it pops out like 25,000 things. So it's very hard to narrow it down for us at that point. So we decided to, after actually half a year, we decided to just dial it back and make it simple. And this is precisely the kind of project actually that you don't want to have in the lab. It grew into this, it was just like a side thing, and then it grew into this thing that it's consuming tons of time. But now it's getting to the point that I'm back down the curve thinking that it's actually could be quite useful. Yeah. So I encourage you to look at a tutorial and so and here's just I will walk through it very, very quickly. Here's just an example that we have done to illustrate how you can use it. So as I mentioned, IBD is a good example. So we basically searched for IBDs, and we found a bunch of studies that are relevant, and we call this a discovery sort of data set. And then even on, like for example, you can see that even though they're on different GSE numbers, so these are geo study accession numbers, and 2 of the studies actually, in this case, contain identical samples. And so we actually have to go back to the authors to make sure that we remove them, because otherwise you don't want to have too many duplicates. So the reason why I mentioned it is because I want to make sure that it's quite clear that it takes effort to reuse them properly. But once you do it, because you have a hopefully burning scientific question you want to use this data for, it's worth it, because once you go through it, you would have annotated these things and it would be in the system, and others in your lab and also in the community can benefit. So we divided them into discovery and validation cohorts, and the end result is actually that we did this really totally blindly, and we got highly replicable results. So we basically did meta-analysis across, in this case, 4 different studies, and then we try to replicate the results in another independent 4 studies, and this is the effect size you see in terms of, so each dot is a gene, and the numbers on the axes are the fold change, basically, of those genes in disease versus control, and as you can see, it's highly replicable, both in terms of genes, but also in terms of pathways, and so on and so forth. And finally, I just want to wrap up. I have 5 more minutes? Or? Okay, good. Another idea we explored was to see whether we can really get this to work at least not in the whole community yet, but at least in the local community. So what we did is we sent out an email to the local NIH immunologist mailing list and proposed that why don't we do a one-day jamboree. Anybody can come as long as they want to try this kind of approach, and we will give them some scientific questions and let's see what we can do in a day, basically. And so what happened? So we would like to. It's both a social and a scientific experiment, and we want to see on the one hand, are there a lot of interest, and do people actually want to do this, and can we actually do this in a day? And the second is the flip side of it. It's if we pose some scientific question to a crowd, can you actually take advantage of that and actually get something done in a fairly short amount of time? Because you would think it's if you have 100 people come, that's 100-person days basically in one day, right? So can we do that actually? So, so that was what I said already. Can research be accelerated by a team of expert volunteers? So these are actually experts. And so the question we pose, it's relatively straightforward. We ask a set of teams to look into human, mostly whole blood or PBMC data sets across 5 different autoimmune diseases. And we ask another set of teams to look at mouse data sets, mostly from tissues, which are the matching mouse models for the human diseases. And then we suggested some specific questions that they need to keep in mind as they gather and put together these data sets. For example, we want to compare common immunological and inflammatory signals across these diseases. And there are also lots of treatments in terms of, especially in the models out there. So how do these treatments really alter the transcriptomic profile within a given autoimmune disease or a mouse model? And the major question is really, if you compare the human and the mouse signatures, how comparable are they, and are some mouse models better compared to the human data or not? So, so what happened over a day of lots of cookies and drinks and so on and so forth? No alcohol, by the way. It's not allowed on the NIH campus. So we formed these multiple groups of 3 and basically divide them into the mouse and the human teams. And then in the afternoon, they came back together and talked to each other. So the matching human and mouse teams talked to each other in the afternoon. And then based on that, they further refine what they gather and all these comparison groups. So here are the statistics of what happened. So 29 volunteers, more came, but some left during the day. 29 actually stayed throughout the day, the whole day, mostly with immunology backgrounds, MDs or PhDs or both, from 13 different institutions. I listed them on the right there. So we constructed overall over 100 of these comparison groups, and the analysis is still ongoing, but it's already quite clear that many of the CGPs constructed are quite high quality, so they're biologically very meaningful as we look through them. And when you look at signatures, for example, across the data sets in humans, there's definitely very strong conserved signatures across data sets that we can detect. And then even when you look at human and mouse, we are starting to see in certain diseases, not all, very strong conservation. So we're starting to analyze that a bit more and see what kind of insight that's going to generate to inform next steps. And so just a summary, so OMIC, it's open and free, and I just illustrated in a day, you can actually get individuals to learn it and be able to start under some guiding questions, start really putting the right CGPs together and start to move towards a biological question. And then they also got to learn how to use the tool in that one day. So I thought that was actually quite an interesting outcome. And the key is really we would like to promote data and metadata sharing in the community to sort of really promote an open culture, so that because these data would just become bigger and bigger, and there's more and more of them. So we have to somehow organize them, and I don't think that's doable by any single lab. So we have to come together as a community and provide ways through which we can start to really put them in a structured way so that they're more usable and reusable. And there's also, I want to mention, a educational mission of doing this. And I've gotten emails from high school students and also postbacks on campus expressing how interested they are about this, because they're in this being educated in this era that biology is really becoming more of a data science, and it's important to be able to really touch the data firsthand. And for the Jamboree, I thought it was an educational experience, and we also gotten some feedback that they found it to be very educational. And I think it's also potentially a good approach to accelerate using public data to generate and test hypotheses. So if you gather a crowd and put them together, maybe not a day, but if you want to be very, very serious, maybe a couple of days, you can actually get something done. So what in the future, looking into the future, what we would like to do, I think this Jamboree concept, together with OMIC, if expression data is your forte, we can really provide a kit. We have the tutorial, we already have the slides, we have templates. We can provide it to other institutions to really help build up a community and accelerate the reuse of the data sets. And we're also thinking about lots of improvements. And if you have interest in doing this together, because everything is open, like I said, it's free. And so, for example, how to provide better ontologies and annotations, how to provide more sharing features and expose an API, and maybe we can even promote developmental community overall. So that would be some of the possible goals. With that, I just want to really thank all the key people along the way. For the Jamboree, Rachel again, she's the clinical fellow who made all the videos, and she led the way organizing really the Jamboree, and it was a very nice experience. Thank you.
Thank you for that really interesting talk. I was interested, you mentioned strong conservation, and I know this wasn't a lecture to present detailed results, but could you flesh that out a little bit between murine and human autoimmune illness?
Right. So far we have done some fairly rudimentary analyses. By that I mean, so we basically went into the human comparison. So we typically gather about 5 or 6. So for most of these, for example, RA, we have about, I think, 7 CGPs. By a CGP, I mean, again, the human disease, typically whole blood or PBMC, versus the healthy controls. So, and then we first look for conserved signatures in the human data set so that it's at least robust across these studies. So these are basically genes that are consistently going up or going down in the human comparisons. And then now, based on that, we have a nice set of fairly robust conserved signatures based on human. And then we do the same thing in mouse, and most of those are from tissues. For example, it may be spleen, they may be joint fluids, and so by default, you may not expect a lot of overlap, because you would expect, unless these cells are really leaking out and so on and so forth, or in circulation, you may not see a lot of direct overlap. But I think the inflammatory processes are what we're seeing, basically. We see substantial, for example, interferon signatures in both the mouse tissues and in the human blood signatures. And then there's also other signatures that we're not sure exactly what they are yet. So we're still thinking about whether those are real or those are but by far, I think the strongest conservation in the human mouse are the inflammatory signatures, broadly speaking. Yeah.
It's really wonderful work on the crowdsourcing and using that in a really nice way, is bringing the community together. I just had 2 questions that are really related to each other. The first one is, do you currently store all publicly available data sets? And follow-up to that is, if there aren't any that you don't store, was that based on like quality control decision making? How do you guys sort of handle that?
That's right. So yeah, thanks for that question. It's a good question. So it's also a tough issue. So we are currently not storing 100%, for example, NCBI-GEO. We cover about 90%, largely because there's a lot of platforms. So by platform, I mean a lot of like technology platforms. So for example, if you use an Affymetrix microarray, there's a lot of different versions, but there's also a lot of like rarely used microarrays like a Hitachi microarray or some early Illumina microarray or some RNA-seq data. Actually, by the way, we haven't incorporated all the RNA-seq data largely because the analysis pipelines, for example, for generating, let's say, it depends on what you get, like RPKMs, and so they're not standardized yet to the point that we feel that we can just grab any raw sequencing data and be able to convert them robustly into a usable format. So we haven't really touched that big it's something that we need to tackle because obviously more and more RNA-seq data are being deposited. So to go back to your question, yes, so the 10% that we didn't cover are mostly these very sparsely used technological platforms like ABIQ, PCR array, and so on and so forth that we didn't find compelling enough for us to go into each one and figure out how to process them properly, basically. Yeah. And then we update them every 6 months so far, and we always leave a buffer of 6 months. So for example, now we will be up to about last December because we don't want to, because if it's a new study, sometimes they update things along the way, and we don't want to deal with that, basically.
Okay, 2 questions. Have you used Geo as your main data source?
That's right.
And so have you looked at Area Express where there's more manual curation, which does some of the work that you're doing?
Yeah, so we look at Array Express a little bit, but we haven't found, again, a lot of like standardized annotations there. But I have to say, I think the overall structure over there seems to be, I guess, a bit more informative overall. And also, I think it's always a superset of Geo. So it's got the European mandated deposits, plus they always update stuff from Geo. So in retrospect, it may actually be a good idea to always sync with Array Express.
And a separate question, we've actually started, so metadata capture by investigators is one thing. There's always going to be errors in there. Some places they have outdated whatever the sample set information. So and then there's simple sample switches that they don't even know about. And
that's right.
So we've been trying to use things like HLA typing and actually gender information to. Gender information is one of the nicest ones. You have expression of genes like exist.
That's right. That's right.
Very determined. So can you have you tried to look that for actually correcting existing metadata and information based on the signatures that you, if you take your gender signature and essentially use that to correct your metadata.
Right. No, that's a great idea. We thought about it, of applying some of these, but again, we haven't. That's one reason I really wanted to get the community together, for example, exposing the API so we can easily plug something in and be able to filter and say provide automatic annotations, for example. So that would be one form of automatic annotation. Yeah.
Very nice talk. I just wanted to know if you have looked at any of the flow cytometry data.
Not yet. So, yeah, we look at flow data in our own work, but no, we haven't. For example, Import has a lot of flow data and also flow repository. That's a biggie to sort of get into. Again, I would love to basically be able to expose this framework or overall approach so that if others are interested in, they can start to plug in and use similar templates, if you call that, to really start to enable other types of data. Yeah. Yes, Ben.
Hi, John. The reason I'm asking this question publicly is because I'm also interested in what other people think. So if NCBI were to do some pre-processing of SRA data, we have about 350,000 RNA-seq runs.
Right.
Would it be most useful to you mapped to transcripts or raw gene counts or to the entire genome?
Entire genome.
Like if we were to use HiSat versus salmon versus like the RNA-seq blast mapper.
I think practically speaking, I mean that's my own personal preference would be to transcripts. Yeah. Cool. I mean as far as expression data is concerned. Yeah.
Okay.
So are you guys doing that?
So we are talking about that in an email.
Great.
So if other people are interested.
Thank you.
Thanks, John. Is your talk up here? That was another good talk. I think we have a lot to think about after hearing that. So our next speaker is Bjoern Peters. He's actually a member of the working group. So Bjoern is Associate Professor at La Jolla Institute for Allergy and Immunology. He's going to talk to us a little bit about his experiences with setting up IEDB. And I teased him last week that his goal should be to help us avoid having an hour-long conversation after his talk. So he's going to help basically show us the light and tell us everything we need to do. So here he comes. He's going to solve our problems now.
Thanks for setting appropriate expectations. Okay, so as was just said, I work on the Immunotherapy database, and I was going to talk today about our experience building this resource and potentially touch on the lessons that can be learned from that for doing something similar for our immune receptor repertoire databases. So what's the Immunotherapy database? It's an NIH-funded free online resource, and what we deal with is cataloging experimentally derived epitope information. Our scope is allergens, infectious diseases, autoimmune diseases, and transplants. And we're dealing with a number of different types of adaptive immune responses, T-cell and B-cell responses, but also MHC binding data, MHC ligand dilution data, which is what we capture. The data is manually curated into the database and submitted by groups directly into the database. And we deal with all kinds of different hosts. So when I say the IEDB is successful, we are basing that on metrics. One of the things is since we launched the newest version of the site, we have a continuous increase in users and visitors per month. It's very easy to cheat with metrics like that. We could pictures from Kardashians on the website and we would greatly increase the visitors. But our bounce rate also decreased at the same time, meaning people actually move forward and go through the site rather than just come there by accident and realize it's not what they thought it was. Another metric that we have is citations per year. So at this point it's about 400. That's 2004 is the last data. That's obviously people actually using the site. Another metric is content. This is the number of papers that are processed. These are the ones that actually make it into the database because they have curatable data. In 2011, we finished curation of the historic backlog. So our scope was everything that started typically in the 50s is the earliest papers that we have. And then we were current, and NIH said, that's great. Now we can cut your funding by 2/3. And they did. And so since then we are being on steady state and curating things as they come in. With at this point over 1 million different experiments, categorizing 240,000 epitopes and over 17,000 papers. And as I said, I wanted to talk about the lessons learned, and as I wrote the abstract, I wrote a bunch of things in here, and what I thought was the good experiences, which are simplicity, ontologies, spreadsheets, observing users, stable identifiers, openness, and manual curation. And the bad things, like registering users, the Google fallacy, XML, powerful query interfaces, undergraduate curators, text mining, and engineers in charge. So I'm hoping I can convince you that this is a good separation of things. So I'm going through my standard slides and kind of talking about what we do in the IEDB, and then I'm always kind of highlighting the lessons learned in this context. So the IEDB, as I said, we have two sources of data. One is literature curation. So we're taking PubMed as the initial starting point, doing like query and document classification algorithms to identify relevant papers, which are then curated. And we have from the Epitope discovery contracts that are NIAID funded, they submit directly in the database. So for the curation process, essentially a curator has to take a perfectly fine paper where information is encoded in figures, in materials and methods, etc., and captured in a structured format like this, where obviously the advantage is then to reuse it. But this is a non-trivial process. So as I said, we are dealing with a number of different experiments, but they share certain aspects, and I'll just break down what we do for T-cell responses. So what you see here is that I should not be a graphic artist. This is supposed to be a mouse that is injected with something. I've been told it looks like a sperm. So the information that's relevant here is what is the mouse immunized with? What is it exposed with? Then also, what is the outcome of this? Does this mouse, is it protected afterwards? Say if this is a vaccination, is it protected from challenge afterwards? And then you take, what cells are you taking from the mouse? Say in this case T cells, which you then test in vitro for recognition of an epitope on an APC. So in this case, you want to capture, what is that epitope? What is the assay readout? Do you use, we saw this morning, proliferation assays, interferon gamma production, these kinds of things. And then obviously for this audience, what is the immune receptor? So to take that, so far, people have typically not done both. There's a split in these communities. People have so far mainly worked on either epitopes or immune receptors, and I'll talk about that a little more later. So now, the data structure that we use, and it's not just for these kinds of mouse experiments, but also with humans and T cell, B cell, all these different experiments, the shared data elements are as follows. We have an epitope that is being recognized. So this could be a peptide, it could be discontinuous residues on a protein, it could be a carbohydrate, etc. And in order for us to know about it, there must be an initial document, which might be a journal article that is curated or an author that submits something. Then if there's an epitope, it comes from somewhere. There's an organism from which it was derived, like Lassa virus, the protein it comes from, it could be a protein complex. Then in order for it to be an epitope, it must be tested somewhere. And so there must be some kind of assay that is being run, measuring a B cell or a T cell response. And then typically the B cells and T cells that are being characterized were derived in some way, say, from a person who had a natural infection, like your Ebola survivors, or you have people like, if you take PBMCs from a normally healthy adult, you'll typically say, okay, I'm testing for flu epitopes, assuming that this person has been exposed to influenza, because if it's an adult, that will have happened at some point. Or you have somebody who's been administered a vaccine or something like that. So there's different types of processes that are used to perturb the immune system that have a specific relevance for immune epitope recognition, and by that also obviously for immune receptor repertoire perturbations. For all these things, we're using controlled vocabularies or much more formal ontologies that allow us to express these actually in first order formal logic, and the arrows here actually mean things, but that's not as important. You can also think of this as just that we're using controlled vocabularies in the database fields that we have. Importantly, this is at this point stable. So in 2015, we added the first new database field after 3 years of nothing. So we believe that the way we are capturing this information is sufficient, and which is, I think, specifically important for these things that are also relevant for the immune receptor repertoire kind of things, and we would like to have that to be interoperable. So our lessons learned were, A, the use of ontologies is great. There's all these people are working in these areas to say, like the NCBI taxonomy, to say these are the organisms that are, it gives an identifier for it. If the data rename the organism, or if there's synonyms for this organism, you're dealing with the ontology. You don't have to come up with your own list of viruses, say, etc. And obviously, NCBI taxonomy is used throughout of other resources, so you're tying your information to other information that's out there. Manual curation. So this process, as I said, we have curated every paper published ever that characterizes an immune epitope, and that was done with a team of curators. There's a lot of people who are saying that this is not feasible. It is very feasible. Manual curation is absolutely feasible, and I would argue the quality of the data you're getting out of it by a human reading the paper and putting it in is far better than specifically text mining. Text mining is extremely powerful if you're doing something like, is protein A interacting with protein B, where typically you're looking through abstracts, going through tens of thousands or millions of papers, looking for co-occurrence of these words or something. That way you can really powerfully mine huge amounts of data and get information out. But if the goal is to have one paper curated accurately, and you're saying that the algorithm has to really understand this paper and capture that information, and I'm not allowing for a 40% accuracy rate, which is fine if you're going through millions of papers. But if you're saying this is what's in this paper, you want to be at 99%. And that's just not, at this point, text mining doesn't do that. Also, most of our information actually is in figures, where text mining routinely ignores. They even ignore tables, where the next most one of my data is in there. Undergraduate curators, we started with this awesome plan. We had in our NIH contract how we're going to hire this army of undergrads that are going to read papers and enter the data. That was a disaster. Reading papers is something that postdocs should do. So at this point, we only have postdoctoral people reading this. It's a hard thing to do. So it's data submission to the IEDB. The IEDB was co-funded with a number of discovery contracts, and they were mandated to submit to the data to the IEDB. So this is somewhat different from what we're talking about here, and that has allowed us actually to be more to demand more of the people that are submitting data to us, because they have to. They're contractually obligated to do this. At this point, there's 285 submissions, which doesn't sound like a lot, but they are huge. So at this point, a quarter of the data in the IEDB comes from these discovery contracts. So this is compared to the entirety of the information from the literature. So that's obviously, these are high throughput contracts, and the amount of data that they can submit to us also typically goes beyond what you would publish in a manuscript and supplemental material, because you're putting your entire data set in there. We do have data deposition open to the general community, and actually the Journal of Immunology has put in their requirements to submit data to us. If people do epitope data, we've actually found at the same time that it can be easier for us to just read the paper and put it in ourselves rather than teaching someone how to submit data to the IEDB, which, as I said, was mandated for the contracts, and where we're demanding somewhat more than you can demand from a casual user. This is the submission process. The users are submitting files to the IEDB. There's an automated validation process which says, does the data look right? But then there's also a curator who actually looks at it and then says it does make sense or not, because you cannot find everything in the automated validation. Specifically, if something biologically is sketchy, it might be that they've misunderstood the submission process. Then there's the curator reviews and approves. There's another set of validation, and the submitter actually gets like a link and can themselves say, at this point I want to have it made public, which is typically after it's accepted for publication somewhere. So our lessons here was, we started with XML and engineers, and then I can consider myself a bit of an engineer, so I can talk dirty about them. So I mean, I started with a physics background when I came there, and then so talking about like how to make very precise ways of formulating your data. And if you're then asking an experimental lab to submit data in this format, it just doesn't work. It is important to work with the kind of electronic artifacts people are used to work with. They're used to working with spreadsheet. You can explain that to people. You can tell them how to fill it out. That works. Again, manual curation, I think, and nearly all the repositories, if you submit something to the PDB, you'll get a person looking at it and saying, no, I think something is wrong or something is missing. It is a painful process, but it is a very useful process. And if you're thinking about what John just presented, for example, the amount of effort that you otherwise have to do later on by all kinds of people that are trying to understand your submission and figure out what was right or wrong. If you have one person looking at it at the time of submission, but you can directly talk to the person who generated it, ideally who was working in the lab, they are the best people to fix the data at that time point, rather than having to later try to, I don't know what your success rate with emailing authors. Typically you email an author asking about like, what was your male versus female. If the postdoc is gone, did the experiments, the chances of getting a response are close to zero. Okay. Yes, ontologies, again, we're using ontologies as a tool also to tell people you should in here put a value that is in the whatever biological process terms, etc. So this works. Querying the IEDB. So this is the homepage when you come to the IEDB, and we are really put front and center the query interface. As an example, I'll just show how to query the IEDB for T-cell epitopes that are recognized in Timothy grass allergic humans. So you specify that query as follows. You're looking for epitopes from Bjoern Peters with this Timothy grass. You're looking for that the host is a human. In this case, you're looking for T-cell epitopes. So you query that asking for T-cell assays. And you're asking that these humans actually have an allergy. So the antigen here is always linked to the disease, so this actually specifically queries for people who have an allergy to Timothy grass. Then you do that and you run your query results. What you get here, then we're kind of operating like a travel website. You do an initial limit to your results, and then those are displayed here, but you can further narrow those down, and I'll show some of that later. But the main results are presented in these different tabs. So this query returns 490 different epitopes, which are the specific peptides that were tested in people, the antigen which they come from. But actually first, here this is just taking the first epitope. I can use this filter symbol to just say, okay, I only want to see data about this epitope. Then you're now showing just this one epitope, and these are the 6 assays in which this peptide has been tested. And you see these have different information. There was proliferation assays run on this epitope. There was an IL-5 ELISpot run on it. There was a tetramer staining experiment running on it. So we're tying together all the different information where this specific peptide has been tested. And as you see, they come from different papers. So this is the way, again, to tie together information coming from different sources. It's actually very interesting compared to what Erica has. So this is post hoc aggregation of data. I mean, Erica was showing how they have one consortium and they can really have a superb standardization by ensuring that everybody works with the same epitopes. Here we have something actually much worse, but still it works. You're aggregating information by all kinds of people who say, I've worked with these peptides. You can look at what the 4 papers were. So there's different groups working on this. And so obviously that kind of also raises the likelihood that this is actually a real epitope if completely different people using completely different methods can show responses to this peptide. Going back again to the whole data set, at this query returned 9 different antigens. You can also look at the antigen level. These are the different Fleum pretens antigens. And if you're clicking at this symbol, you're actually aggregating the information together. Typically what happens with epitopes is people have slightly different variants of peptides, somewhat longer, somewhat shorter, and then we're actually plotting here back the response frequency of T-cell responses against peptides derived over the sequence of the antigen. Aggregation of data, I think, is also key. When you look at it, as I said, you have a 1 million experiment, you had your 500 something different epitopes returned. You want to somehow summarize that data, and that's something we're really working a lot on, because just giving you a list of 500 different assays is not really a very useful bit of information. So we have much more detailed information than what we show in this initial query, and you can get to these specialized searches. If you go to the B-cell assay details list, you're getting a detailed search pane, and each of these panes extends further. I just wanted to demonstrate one thing. If you're going for the host specifically, you can say, okay, I'm looking again at humans, but now humans that have gone through one of these immunization processes where the immunological history involved administration with a protein. And okay, now I'm getting back all antibody epitopes that have ever been characterized by humans that have been immunized with a protein. So what will you find? This is the summary of that on the protein page, and what you see is you get hepatitis B virus, Clostridium botulinum, Bacillus anthrax. These are all the licensed protein-based vaccines, which in the end, well, that's not a big surprise, but actually that's something cool to be able to pull those things out immediately. What is it actually that people have immunized humans with on a protein level? And interestingly, and I love this, Clostridium botulinum you might not think should be at the very top of this list, but it is, because these are all the Botox people. So you actually can get antibodies from them. This is another way of where people are actually exposed to something. Yeah. And they actually do get immune to it. So you have to increase the dose later and later, because you actually have neutralizing antibodies against Botox. It's cool. So anyway, lessons learned. So starting with engineers in charge. So when we started off with this, we went through a software design process when we designed the first query interface, having a systems requirement specification document of over 2,000 pages, going through UML modeling of the use cases of how you interact with this database. And then we had the first output of this, and it sucked. And then we said, well, why do you have to do this and that? And I said, well, that was in the UML diagram, so and so. I said, no, that's not what you meant. So it is a horrible idea to try to design a system from start to finish on paper and then come back a year later and look at what the programmers give you, because they will have a very different, there's ways to screw these things up. So what we found much more useful is actually observing users. So we have now gone through a much more rapid cycle of designing, looking at people who are trying to query with your interface, and you're looking there and they're saying loud what they're doing. It's like, and I'm now looking for, where do I put Timothy grass? And you're like, it's there. It's there. Why don't you see? It's very frustrating, because you have to be quiet. But it's very, very informative, and it doesn't require large studies. You have 5 people there. They all make the same mistakes. It's very repeatable. Then simplicity. Again, we had this more complicated query interface. What I showed you first was our default. Nobody understood it. People went to the website and they left immediately. So you have to give people something very easy and simple to quickly get some kind of results. Then you have them captured, and they'll see that they want to do something more complicated. Then you can give them the advanced query interfaces. You want to start people off giving them success. And again, ontologies are extremely useful for the queries, because you don't have to type exactly, if you curate at one level, and then you can query at a level above because you have in the ontology actually not just synonyms but also a parentage hierarchy, so you can query for, say, I don't know, influenza viruses and you get all the different isolated back, or you can go higher and just query for viruses and you get influenza next to hepatitis B, etc. Yeah, powerful query interfaces, as I said, they have their place, but they are for power users, and your power users are going to be yourself and a few % of your user base. It's much more important to focus on getting the simple things right. The Google fallacy. Yes, people routinely say, your interface is too complicated. They'll always say that. And then they say, why aren't you Google? I want to have one line and just type your data and that's great. But it does not allow you to do ever a query like this, where you're saying, I want to have things where the person was immunized with a protein. If you have any kind of text search, it'll confuse that with things where people were tested for recognition of a protein. You cannot, unless you derive a very, very, very complicated text search, you're not going to have that power. The point of curation is to have structured data, and the query interface should take advantage of that structured data. If you want to take advantage of a full text search, and you think people should be becoming experts of regular expressions and logical parameters, that's cool, but then use Google. There's no point of curation. You can just put the full text in there. And that actually is fine. It works. Okay, so these are the things I didn't manage to cover. So registering users, that's what we first did. And there were some concerns that the data that would be captured would be enabling bioterrorists because they used the epitopes and because we were funded by the biodefense money. But what we captured is the same data that's in the papers. So we did actually manage to say, well, if we are supposed to be under lock and key and you have to register for it, why is that same information available in Journal of Immunology? So that works. The thing that's really happened with registering users is we look at the dropout rate. The moment somebody has to fill in their email address, more than 90% leave the site at that point. One. Secondly, it'll not be on Google. Meaning if somebody wants to search right now, you can put in a peptide string on the peptide that you have looked at, an IEDB page will pop up giving you all the information about that peptide where it was tested. If you file registration, Google cannot index it, will not give you that page. You're losing a vast majority of people that come to the site. Stable identifiers, it's extremely useful because you want something that's called one thing at some point and something later at another. You want to be able to change your labels while keeping an identifier intact if it's the same thing. Openness, this actually goes with registering users. We're not trying to protect our data. We are giving away you can download the whole database and do whatever you want with it. And we found that that helps us. That enables other power users who want to do things that you can't easily enable to just do that by themselves. So now specifically for the immune receptor data in the IEDB, so currently we do curate antibodies in TCR sequences if they are known to be epitope specific, because we are tying everything to an epitope, otherwise it's not in there. So until recently, the vast majority of data that we have where a receptor is known is X-ray crystallography, because obviously there you do know the sequence of the antibody, say, that you crystallize, or monoclonal antibodies, or TCR transgenic mice. Now we have an influx of receptor data that comes from things like tetramer sorted T cells, or you have sorting by antigens that you're tetramerizing for B cells, or you're doing proliferation experiments or something, or you're doing based on statistics, you're saying that these must be epitope specific. So that's actually a new focus for us to make. We have receptor data, but it's currently somewhat hidden because it was in just a very rare fraction of the data. So we are now going to be adding a receptor tab here, so you can run the same kind of query and then you get your receptors back. So similarly, so we currently actually have a dump of all this receptor data, so you can download it on the IEDB, but it's not in a very useful format. We are trying to get this better and make it on the main page. More specifically, you can imagine, like in the example I showed here, when you're looking at all the data where this peptide was tested, in all these different experiments that were run, if somebody had tested the receptor, you could have that receptor data here living side by side and saying, okay, in this paper, somebody had a given TCR that recognized this epitope, and this was from a person who had allergy to pollen, And it's a class 2 restricted epitope, and the T cells that express that receptor and recognize this peptide were proliferating once stimulated. So it's, to me, the way this part of the information is structured is very, very similar in the IEDB, and that's why we would love to have interoperability. So we are currently not funded to do full repertoire sequencing, but as an obvious disclosure, if there was a funding opportunity, clearly we'll apply. I would be lying if I was saying or not, and so if you want, you can say, forget everything I said and say I have a conflict of interest, but you can also say I am just very interested and passionate about the subject. So I think everybody here should hopefully have some kind of conflict of interest because we are all interested in this. But obviously, whoever gets this, we want to be interoperable with all the resources out there. So we will try to comply in terms of any kind of data standards that are derived from the standards working group, how the sequencing information, which we currently don't capture at all, how did you get to the antibody or T cell sequences in the same way in the IEDB, or even better, we would love to have a link for a given epitope. We're saying these are the T cells recognizing it. Ideally, someone else has the repertoire sequencing data for those T cells. And it would be nice if there was a link back to say if there's, at the moment you have epitope-specific repertoire data, it would be nice if that could be linking back to the IEDB. Yes, and as I said, we actually are already working on the other way, so we would love to see the same way we are capturing this, like this is an allergic individual kind of information, to be captured the same way, because it is very, very, very similar data, and it's going to be very powerful to link these things up. Bonus points. I was reminded yesterday, there was a number of talks about data quality. We've gone through that for the IEDB a lot. And people were saying you shouldn't capture all the epitopes, only capture good epitopes. And everybody agrees with that. But what is a good epitope? Somebody will say, if you don't have the 3D structure, how the antibody binds with the antigen, you don't know the epitope, and that's true. Others would say, I do not care about antibodies recognizing some circulating whatever things. The antibody has to be neutralizing. Or somebody might say, your assay isn't actually giving us meaningful binding data. It should have a certain affinity threshold, otherwise it shouldn't be captured. The allergy people saying, what are you even talking about? T cell epitopes. Everybody knows in allergy the only epitopes are IgE antibodies binding protein. Other people are saying, why do you have all the mouse data? You should only do human because you're confusing people. In T cell data, people are saying it doesn't, if you get an in vitro expansion, that's an artifact. You can also only report things that are direct ex vivo. Then people were saying, oh, you have these 20 peptides in there, but we know the class 1 epitope is 8 to 11 residues and it needs to be mapped to the degree. Or if you don't tell me the HLA restriction of your epitope, so all these things are valid points. And for different users, it might be completely useless if you don't do any of these things, but you have different users. And so in our approach has been, we capture all the data. If it's published, that's our quality control. If it's a published article, the data goes in, and then the users should say, in my case, I only want to see human data. I only want to see, so this is, we are viewing ourselves similar to PubMed. PubMed doesn't turn down the abstract because it has a spelling mistake in it. It lets the users decide if people make a typo in the title, if they want to trust that author. But on the contrary, we're trying to have reasonable defaults to allow default searches that are giving you quality data back. But it's somewhat hard, or at least we're prioritizing things in the search results that have more support by being in more papers by different groups and more assays. So we're trying to have a mixture there that is a measure of quality in terms of what we report first. Finally, dealing with multiple repositories. In the case of the IEDB, there is a second and older pioneering database called the Los Alamos HIV Immunology Database, which is dealing with HIV epitopes, and they are completely incompatible with what we are doing for all the other infectious agents. And this is a result of different funding agencies like DAIT, D8s, and it is completely understandable. From a funding agency's mission, they're going to say they have all the HIV money, they have so they should be paying themselves for this. And then NCI has a completely different funding pot again, and you cannot force funders to collaborate, and they will have different priorities also. So another agency might be coming up with you should a different priority, different approach. So here we don't have this yet. it would be awesome if we're doing this kind of upfront standardization that we talked about, to have interoperable ways of representing our data, to have interoperability. We can say that we want to have one repository, but there's political and social aspects that are going to make this potentially impossible. So let's try to prepare for a future where there can be distributed repositories, but as a bioinformatician, I don't care. I want to get all the epitopes, say that I have a crystal structure for them, and if one of them is funded by someone else, I still want to see it. So this is my last slide, or one before last. So the IEDB is an example in my mind of a successful repository of immunology-related information. We had to go through multiple redesigns, and we did many errors to improve data population, query, and reporting. So some of these are, I think, lessons learned that are relevant for designing AIRR repositories, and we have a strong desire to keep any of these repositories linked to what we're doing in the IEDB both ways. And obviously this is a team effort. This is our curation team. This is Leidos, who are doing much of the database hardcore handling, collaborators in Denmark, and postdocs in Mailand. Thank you.
So we're running a little bit late. Maybe just a couple questions, and then I don't know. We want to get an hour in. We've got a good long lunch break. We want to get an hour discussion in. So maybe we go right into the discussion.
Just a quick question. What does the stack look like in terms of the implementation?
Stack.
What are the
database backends? So we started with an Oracle database. That was a disaster. Because then you can't give it to anyone. So we actually switched to MySQL implementation, running in the PHP website above it. Is not technically fancy. We found that to be much better for, we started with like a whole Java 2EE fancy, technical, complicated thing where then it was a huge turnaround time to do little improvements. So we now have a hacky implementation of a PHP-based website over MySQL, kind of a LAMP stack.
This is a question from a remote participant, Nishant Matharadan. Have you explored crowdsourcing solution for manual curation by experts outside of your group in the field by common systematic user interface or tool?
Of the user? Sorry, I didn't get the last
part. I think the idea is that could you expose an interface by which experts outside of your group could perform manual curation?
That's a very interesting suggestion. We have not explored that in detail. Primarily, we've been relying on experts submitting us something. So we are always saying, and actually we had we have workshops where essentially in the field, say we've finished curation of one area, say allergy, we invited a bunch of allergy experts, gave them the data, and then they provided feedback. So we are asking for essentially people to submit a bug report. That might not be optimal. Obviously, adding some more user annotation might be good. Yeah.
For the ontologies, did you find that you had sufficient coverage of the immunology domain? Are there gaps that you know of?
There are plenty of gaps, and we're actively working. Essentially, if you want to be a user of ontologies, a serious user of ontologies, you have to be a developer of ontologies, because essentially we are actively contributing to a number of projects to ensure that the terms we need are in there. We have a mechanism where we are creating preliminary terms where we're assuming they're going to be in ontology at some point. In some cases it's taken, Lindsay knows about this, a very, very long time, and they're still in our preliminary data. So there's definitely gaps. There's some areas that are extremely well covered, others not.
Could you tell me which ontologies that you're working with?
Okay, this is going to take a long time. So for the experimental characterization, OBI, we are working with, no. What do you have it? Yeah. So I mean, NCBI, Uniprot, KEBI, OBI, Go, IAO, DO, IDO. Those are parts, but there's a bunch of other ones: cell ontology, protein ontology. There's an MHC restriction ontology. Yeah, a whole bunch.